Okay, good morning. I think we're going to get started. So thank you everybody for attending. This is the UN International Day of Women and Girls in Science highlight on sleep research webinar. So today we have 4 panelists who are going to talk not only about their experiences as women in the sleep research field, but also as about the importance of sleep and gender as a research covariate in sleep research. All right. Before we begin, I have the mission statement from the ESRS Gender Equality Forum, who's hosting today's talk. My name is Jaime Devine. I'm on the GEF. So the Gender Equality Forum is a task force for both early career and senior scientists of any gender identity that aims to raise awareness about the concept of gender inequality, identify current issues and attitudes regarding gender inequality within the society, and promote equal treatment of all European Sleep Research Society members. Before we begin, I have included a brief list of definitions that may be relevant to today's talk, but also to understanding the goals of the GEF. First is sex. Sex is a biological category based on reproductive, anatomical, or genetic characteristics. Compared to that, gender is a socially constructed concept about the roles, behaviors, activities, or attitudes that should be associated with a given sex. Gender identity is a person's own identification of whether they are male, female, non-binary, etc. And that can be in line with their biological sex, or it can be different. So everybody does have some sort of concept of a gender identity, even if it is a man who was born a man and does not have any problem with that. And lastly, gender inequality is the idea that sex or gender changes somebody's ability to have access to rights or enjoyment of those rights in a societal way. So those are the definitions. I will stop sharing my screen and introduce our first speaker. So our first speaker today is Adriana Michalak. She is from the MOMI Lab Research Unit, Sleep Plasticity and Conscious Experience Group at the IMT School for Advanced Studies in Lucca, Italy. Her talk today is Significance of Addressing Sex Differences in Sleep and Cognitive Research. Dr. Michalak is a cognitive scientist working on sleep research since 2014. Currently, she is a postdoctoral researcher in dreams engineering at the Space Group at the IMT. Since September 2022, Adriana is an elected member of the ESRS Early Career Research Network, where she plays the role of sustainability lead.
Okay, thank you very much for introduction, and I would like to start with thanking the ESRS Gender Equality Forum for organizing this webinar and the invitation. So my current scientific interest is related to dream engineering, but today I'm going to focus on a work related to my PhD project that was conducted at the University of East Anglia. In a nutshell, my PhD thesis focused on the interrelationship between sleep, rest activity patterns, and APOE4 genotype that is the strongest known genetic risk factor of Alzheimer's. One of the research questions that we asked and that has really changed my scientific mindset was how biological sex modulates the effect of sleep restriction and time of the day on vigilance and recovery sleep in the healthy elderly men and women. Our study consisted of 4 phases, that is recruitment, screening session, actigraphy session, and the sleep laboratory session. And today I will focus just on the sleep laboratory session. During our lab sessions, we ran the total sleep deprivation and the multi-nap protocols. Both of our experimental protocol lasted for 2.5 days, and both of them started with the baseline night and both of them finished with the recovery night. Throughout the time of the experiments, participants stayed in the sleep research unit in the dim light condition. In both protocols, we have assessed cognition and mental effort on 4-hour basis. Today, however, I will focus only on subjective sleepiness assessed by Karolinska Sleepiness Scale and vigilance assessed by Psychomotor Vigilance Task. And that is a basic reaction time task. What is more, in the multi-nap condition, we have scheduled 9 80 minutes long naps every 160 minutes. So basically, in the sleep deprivation protocol, our participants were awake for 40 hours, so their sleep pressure was constantly increasing, whereas in the multi-nap condition, due to scheduled short naps, we aim to keep the participants' sleep pressure at the constant-ish level. To tell you a bit about our results, over here I have got the Karolinska Sleepiness Scale results and PVT results. On the x-axis, you can see the time spent in the protocol. And on the y-axis, I have got KSS scores and reaction time scores. Both of them are expressed as ratio to the first cognitive assessment, that is assessment right after the baseline night. Okay, so importantly, time spent awake was associated with gradual increase in subjective sleepiness and also decrease in objective vigilance in the sleep deprivation compared to the multi-nap condition, which suggests that our sleep pressure modulation protocols worked as intended. What is interesting, we found that vigilance was more affected in women in sleep deprivation condition, but also women, they benefit much more from scheduled naps. So the difference between a high sleep pressure condition and low sleep pressure condition is really significant. We have seen also a trend where women were reporting higher level of sleepiness throughout the protocol, the sleep deprivation protocol. But what is the most interesting results for us was what happened with the sleep architecture during the naps. So to start with, the participants were comparable during the baseline night, besides female having significantly more non-REM free sleep compared to men. So it's something that I need to disclose. And what's important, sleep parameters across the naps showed expected circadian pattern with longer total sleep time and sleep efficiency and shorter sleep latency and more slow wave sleep during the circadian night. So for example, over here, you can see that slow wave sleep was the longest during the first part of the circadian night. But what was super interesting to us was that even though that men and women have similar total sleep time, sleep efficiency, sleep latency, women tend to spend significantly more time during in the deep sleep, whereas men spend significantly more time in N2 sleep. And it was, in case of deep sleep, we could see it across all the naps. So it was very intriguing results. And here comes my biggest regret, my biggest PhD regret, which makes my presentation kind of cautionary tale, because we have not had specific questionnaires asking about menstrual cycle, menopause, or andropause, which would help to put our results in better context. Okay, so to conclude, we showed that previously reported sex differences in non-beneficial effect of sleep loss on objective vigilance in younger people are present also in healthy older people, because previously women were shown to present increased vulnerability to sleep loss compared to men. So right now we also showed that in elderly adults. And also our results suggest that biological sex should be considered when we are studying effect of sleep impairment, jet lag, or shift work in older age group. And also differences in recovery sleep architecture should be addressed in the future, for example, when one is assessing effects of chronic sleep restriction among shift workers, for example, nurses, and when one wants to implement sleep compensatory interventions, for example. And last but not least, as I mentioned, I focused doing my PhD on Alzheimer's disease, and there is growing body of evidence suggesting that female sex is suggested to increase the risk of Alzheimer's. And we know that there is this well-established bidirectional relationship between Alzheimer's and sleep and circadian alterations. However, these interrelationships, they are not well understood, which is yet another reason to treat biological sex differences seriously and pay some attention towards that. Okay, and at the end, we have been told that we can share some of our personal experience as women and girls in science. So on personal note, I have been thinking a lot what to say, but yeah, I have grown up in post-communistic patriarchal Poland, and throughout our school education, we have been told that math, physics, engineering are subjects for boys, while languages and humanistic subjects are for girls. And I used to believe in this imposed binary cognitive frameworks until in high school, my amazing math and Polish teachers, they made me understand that I don't need to fit in these gender-specific cognitive boxes, and that those boxes are fake, empty, and outdated constructs. And these early educational experiences, they made me realize how strongly the imprinted social constructs can influence faith in our academic abilities, in our self-esteem, and consequently drive our career choices. So today, I would like to wish all the girls and women in science to do whatever they want and to do science without the limiting shadow of traditional gender roles. Thank you for your attention, and thank you everyone involved in the study that I have just presented.
Thank you very much. That was a wonderful presentation. We're going to hold all questions till the end of the seminar to allow all of our speakers time to talk. If you have a question, please put it in the Q&A, and we will read them aloud, or you can ask them yourself. Please indicate if you would like to ask it yourself so that we can unmute you. Our next speaker is Dr. Brooke Aggarwal. She's from the Columbia University Medical Center in New York, New York. Her talk today is Sleep and Cardiometabolic Risk, Sex and Gender Influences. Dr. Aggarwal is a behavioral scientist, clinical health education specialist, and assistant professor of medical sciences in the Division of Cardiology at Columbia University Medical Center in New York City. The focus of her research program is sleep and cardiometabolic risk among diverse populations of women at various life stages, including childbearing years and the menopause transition. She completed her doctoral degree from Columbia University, a master's degree from the George Washington University, and an NIH postdoc research fellowship in preventative cardiology from Columbia University. She served as principal investigator and director of training of the American Heart Association funded Go Red for Women Center at Columbia University, and designed sleep and cardiovascular health projects among women. Thank you.
Thank you so much for having me today, and thank you for the kind introduction. I'm very excited to share with you some of our research on sex and cardiometabolic risk and the ways that sex and gender can influence that research. So in the United States, the red dress symbol has come to symbolize increasing awareness of heart disease as the #1 killer of women. And surprisingly, only 43% of women actually know that heart disease is their #1 killer. And women also are underrepresented in clinical trials of cardiovascular disease. So only 39% of participants in clinical cardiovascular trials are women. And today, what I'm going to be talking about is that poor sleep contributes to increased cardiometabolic risk among both men and women, but women actually may be more susceptible to the negative effects of lack of sleep. And I'd also like to share that a woman's life stage, for example, menopausal transition or childbearing years, may impact the associations between sleep and cardiometabolic risk. I like this slide. This is from Dr. Clayton, and it shows that sex and gender are important to take into account for cardiovascular health at all levels, including preclinical research all the way through to health policy, because there are sex-specific cardiovascular risk factors that would inform treatment. For example, women often present with non-specific or unusual symptoms of heart attack. So we often think of heart attack as clutching the chest, but in women, surprisingly, sleep disturbance 1 month before a heart attack is very common. So about 48% of women experience that as a symptom of a future heart attack. Of course, sleep duration is linked to increased risk of coronary heart disease. So there's a U-shaped curve, and the sweet spot seems to be between 7 to 8 hours per night is considered lower risk for heart disease. My colleague Norm Akram and I were interested in studying gender differences in the associations between insufficient sleep and cardiovascular disease risk. And so we conducted a review of the literature. And the take-home point here was that women may be particularly vulnerable to the effects of poor sleep on cardiac health. And in 2016, our colleague Marie-Pierre Saint-Onge at Columbia University was the chair of the first Heart Association scientific statement on sleep duration and quality and its impact on cardiometabolic health. And one of the research gaps or priorities that was identified was the importance of inclusion of women in diverse populations in trials on sleep and cardiovascular health, because they may be more prone to sleep disturbances than men. Women may be particularly at risk for poor sleep due to lifestyle factors such as caregiving. They also may have mood disorders and may be more prone to depression and anxiety, as well as taking into account their changing reproductive hormone levels after pregnancy, during the menopausal transition, for example, that may affect their sleep. And so in 2016, my colleagues and I at Columbia University started the American Heart Association Go Red for Women Sleep Center at Columbia University. And the goal of this center was really to build the evidence base around sleep, to establish it as an official risk factor for cardiovascular disease, and try to incorporate sleep screening as part of routine clinical care. This was one of the studies that we published on sleep and cardiovascular health. It was a collaborative study, and not surprisingly, we did find that poor sleep was associated with increased blood pressure. So here we show that poor sleep quality was associated with higher systolic blood pressure, and risk for obstructive sleep apnea was also associated with higher diastolic blood pressure among women. My colleague, Dr. Michalak at Columbia, was able to directly characterize the vascular endothelium. And interestingly, what we found was that nuclear factor kappa B, which is a pro-inflammatory transcription factor, was associated with greater insomnia severity, as well as poor sleep quality and longer time to sleep onset among women. So as part of our findings, we concluded that these common but frequently neglected sleep disturbances, such as poor sleep quality, insomnia, were associated with increased endothelial inflammation. And the key was that this was in women who had adequate sleep duration. So we were measuring their sleep duration by actigraphy, and they were all sleeping 7 to 8 hours per night. But if they perceived that they had poor sleep quality, they actually did show increased endothelial inflammation. One other study that we published from this work was an analysis of poor sleep patterns with overall cardiovascular health among women. And the key finding here was that poor sleep quality, higher insomnia severity, snoring, and higher risk for obstructive sleep apnea were associated with a lower overall cardiovascular health score among women. And then when we stratified by menopausal status, we actually found that these associations were stronger among postmenopausal women. So 20 years ago, we had to take action to increase awareness of heart disease as the #1 killer of women. And now we also took action to show that sleep should be considered an official risk factor for heart disease. And we're very excited that in June of 2022, the American Heart Association adopted sleep as an official 8th metric of heart health. And so now women and men are screened for sleep as part of routine clinical care. And one of the things that I think is very important is that as we're collecting this data, we continue to take into account sex-specific factors when we consider both sleep and cardiovascular health. So I'd like to acknowledge all of the wonderful investigators that contributed to this work. And thank you for your time.
Thank you very much. We'll go on to our next speaker. Again, if you have any questions, please put them in the Q&A and we'll answer them at the end of all 4 panelists. So our next presenter is Dr. Jessica Mong. She is from the Department of Pharmacology and the program in neuroscience at the University of Maryland School of Medicine. Her talk today is Understanding Estrogenic Mechanisms Governing Sleep. Dr. Mong is a professor of pharmacology at the University of Maryland. Her research interests have been centered around biological sex differences and steroid actions on brain function in the overarching theme of contributing to the betterment of women's health. Dr. Mong established an active and exciting research program investigating the effects of ovarian steroids on sleep and arousal using both rodent and non-human primate models. Her work has demonstrated that sleep patterns in females are more sensitive to fluctuations in sex steroids compared to men, and that sex differences in sensitivity is the result of sexually differentiated neural patterns in the brain, in the sleep circuitry. Her research serves as a platform for disseminating the importance of understanding why women are more at risk for sleep disorders throughout their lifespan.
Okay, thank you for that introduction. It was a great overview of what I'm about to talk to everyone today about. And I also want to thank the organizers for this absolutely fantastic webinar and an opportunity to discuss sleep and women. So I just want to start to give us a sense of really I think, sex and potential gender bias in sleep disruption between men and women. And so this is a sort of copulation of data from both longitudinal as well as meta-analysis studies. And what this demonstrates, and this is just representing the percent occurrence of insomnia symptoms in women starting during childhood and going through out the lifespan. And I just want to draw your attention to the fact that around puberty, and especially around the perimenopausal menopausal period, we see that there's a significant increase in the risk for developing insomnia or sleep disruption in women and boys compared, women and girls compared to men and boys. And we've known this for some time. But perhaps what we don't think about, although I think we've already heard today that there is a significant risk factor for women, is the fact that this insufficient sleep actually puts women more at risk for not only the cardiovascular disease related mortalities that we've already heard about, but actually anxiety related affective disorders such as major depressive disorders, as other and other mood disorders, breast cancer, metabolic syndrome, and obesity. And so what I find most surprising is that despite the fact that we have this bias towards women having insufficient sleep and risk for insomnia, and that the consequences of this to overall health, we really know surprisingly little about how biological and sex and gender actually influence sleep. So one thing that we do know is that based on a number of clinical as well as preclinical studies, is that disrupted sleep in women is often associated with changes of ovarian steroid profiles across the lifespan. So if you think back to the graph I showed you in the beginning, that is at times of puberty, of course, across childbearing years, and especially at menopause. And so when I entered into my independent research career, one of the questions that I was really broad questions that I was interested in asking is that what drives these sex differences in sleep disruption that are reported in the literature? And of course, you can always start by answering a question with questions, right? So sex steroids is one possibility. In fact, there's evidence to suggest that sex steroids are playing a role. But what about just basic sex differences? And getting back to the slides that were first presented before we started, that is it something that's chromosomal, just actually biological sex and not necessarily related to hormones? Or, of course, when we talk about women and men, we can exclude the cultural, societal, and gender-related demands that are placed upon women. Now, I am a basic science researcher, and my primary model is the rodent, particularly or specifically the rat. And so for us moving forward in my lab, we really can only address two of those three questions, and that is sex steroids and basic sex differences. And so one of the very first studies, well, and I should, before I get into that, the way we do this in rodents is very similar to the way we would do it in humans. And that is we actually use a telemetry system that allows us to collect the EEGs and EMGs and allows us to actually stage and score the sleep cycles across the rodent sleep-wake patterns. So the first question that we asked was, are there just basic sex differences in sleep behavior? And so to do this, what we did was we removed all endogenous steroids by castrating males and ovariectomizing females. And once the steroids had cleared from the system, we essentially just measured their sleep-wake patterns across a 24-hour circadian pattern with light and dark cycles. Equivalent light and dark cycles. And really, essentially, what we found in this study is that in the absence of sex steroids, we could not detect a difference in the way male and female rats were sleeping in their behavior. Now, this next slide that I'm about to show you is really a compilation of work that took about probably 5 years in our lab. And what we did here is that if it wasn't based on basic sex differences, that is without the hormones, we really tried to dive deep into then what is going on in terms of the steroidal milieu. How are estrogens and androgens affecting sleep in male and female rats? And so what we basically showed through these studies is that when you ovariectomize a female and you give her back physiological levels of estradiol, you can see changes in her sleep patterns up to about 50%. When you gonadectomize a male and you record his sleep and you give back testosterone and estradiol, you barely see maybe about 10% of changes. And most times this is not significant from the non-steroidal baseline. And so our major findings throughout these studies is that the major sex difference between males and females, at least in the animal world, is that it's the sensitivity to the changing steroidal milieu. And the way I like to think of this is that the female brain has remained somewhat plastic in terms of her sleep circuitry, whereas the male brain has become a little bit more permanent or cemented and not conducive to reacting to these changes. And in fact, if you dig into the clinical literature and you look at men and women's sleep patterns over their lifespan, the clinical literature somewhat supports these findings. So why is sleep in females then more sensitive to the effects of estradiol? Again, using our rodent model, we were able to build off of a very classic paradigm. So we know in the mammalian world, whether it is rodents or non-human primates or humans, we know that sex differences in the brain are established during an early developmental period, which is what we sort of call a critical period. And what I'm showing you here is this critical period for a rat. And so essentially prior to birth and just after birth in rats, there's this tremendous surge in males of testosterone. And this surge in testosterone actually is has a programming effect on neural circuits. And the female has an absence of this major surge of gonadal hormones. And so she does not have the same sort of organization of the brain as the males do. And so what happens, this is happening developmentally, but then what happens in adult behavior, these pre-programmed circuits, when the adult's hormone levels start to turn on and circulate, the testosterone will activate what we call male-like patterns in a biological sex term, and female-like patterns when estradiol is turned on. And you can think about this in terms of physiology, such as reproductive physiology as well as reproductive behavior in the rodent. Now, interestingly, during this sensitive period, the brain is really bipotential, meaning that we can actually take a female, treat her with the same levels of testosterone, and we can reorganize her circuits to be more like a male. And so we did this for sleep. And what we found, and we asked the question, is sleep behavior and its neural circuitry programmed by the effects of early exposure to steroids? And in fact, what we found was this, and I'm just referring to this group of females that received testosterone because their patterns were more male-like as masculinized females. And so again, we see that in normal females, we see that change of sleep patterns as a result of estrogen. Males are not changing their sleep patterns when given the same dose of estrogen, and neither are the masculinized females, suggesting that this pre-programmed effect of steroids is affecting the sleep circuit. And to go a little bit further, we actually delved into identified sleep nuclei, and we looked at the activation patterns of what we call sleep active neurons. And in fact, the activation pattern of these neurons correlates with the behavior, where we see that the males and masculinized females have significantly fewer activated cells than the normal or the control females. So my slides have frozen. Sorry about that. So just to summarize then our studies, the female sleep circuits we've shown are more sensitive to changes in adult steroidal milieus, and that the sleep circuits are developmentally programmed by early exposure to sex steroids. And there's evidence in clinical literature that this may also be the case. And that moving forward, and what I don't have time to talk about today, is that our current work is investigating how estradiol is affecting the cellular and molecular components underlying sleep. And sort of why is this important? And I'll leave this as my last thought, and that is because historically, treatments and pharmacology have been generalized to the male physiology. And understanding the role of estrogenic action on the sleep circuitry in the female brain actually may aid in improving interventions for treating sleep disturbances as women as well as men, because it helps us understand the circuitry better. And so I just want to end with my students and postdocs that have contributed to this work, as well as my funding sources. Thank you.
Thank you very much. I will introduce our fourth speaker. Dr. Maria Hrozanova is from the Institute of Public Health and Nursing in the Norwegian University of Science and Technology in Trondheim, Norway. Her title is Sex Differences in Social Jet Lag. Dr. Hrozanova is a postdoc. She gained her PhD in 2021, investigating the associations between stress and sleep in junior athletes. Currently, she's a project leader of a nationwide pragmatic multi-center randomized controlled trial investigating the effectiveness of group-delivered cognitive behavioral therapy for insomnia in Norwegian primary care. She's our fourth speaker. Again, if you have any questions, please put them in the Q&A and we will answer them at the end. Thank you.
Thank you so much for that introduction and for the kind invitation to present today. I would like to take this opportunity today to talk about a recent paper on sex differences in social jet lag that me and my co-authors that are pictured here have submitted to the Journal of Sleep Medicine. So, firstly, a little bit about what we know about the sleep of teenagers. We, of course, know that sleep plays an important role in many of the formative developmental processes that occur during adolescence. However, at the same time, there are multiple things happening at this age which bring about an evening shift in alertness and preference for later bedtimes that we see among teenagers. And these include biological factors, such as changes to the sleep-wake homeostatic process and circadian regulation, as well as psychosocial factors, such as exposure to evening light from electronic devices and early school start times. And we see that this evening shift contributes to chronically reduced sleep durations among teenagers, which has been previously described as kind of the perfect storm of short, ill-timed, and inadequate sleep among teenagers. One actigraphic study has looked into the habitual sleep of teenagers and found that only about 10% of teenagers gain the recommended 8 hours of sleep during the weekdays. And during the weekends, this proportion increases to 40%. But it is this discrepancy between sleep during the week and during the weekend that is called social jet lag. I think everybody who has met a teenager or who has been a teenager themselves knows that, and research confirms this as well, that social jet lag and sleep loss more generally have a very real influence on teenagers' lives, impacting everything from their energy levels, motivation, and academic performances. But surprisingly little research has been done on this topic, especially utilizing objective measurements of sleep. And I want to emphasize this because we know that subjective measurements of sleep, like questionnaires or sleep diaries, they capture a different dimension of sleep than studies done with objective measurements, such as polysomnography or actigraphy. We know, and Jessica has talked about this as well a little bit, we know that girls and women report worse subjective sleep quality than boys, and also there is a higher prevalence of insomnia among girls and women. But measured objectively, in a lot of studies, we see that girls' sleep patterns are actually kind of better than those of boys. And there is quite a bit of research on social jet lag using subjective measurements of sleep, specifically most often sleep diaries. but only 2 existing studies have utilized objective measurements, and more specifically actigraphy. And from these 2 studies, we know that girls actually sleep more and go to bed earlier on weekends than boys, which fits kind of in this with this greater kind of picture of this discrepancy between objective and subjective measurements of sleep and sex differences in sleep patterns. But since the existing evidence on social jet lag is so limited, we were wondering how to kind of go further from that. For instance, sample sizes in the previous studies were rather small, and they also pooled all weekend and weekday nights together, kind of obscuring a more detailed understanding of what happens on the day-to-day with the sleep of teenagers throughout the week. Also, no previous research has investigated whether sleep stages are also subject to sex differences in social jet lag. And that brings us to our study, in which we looked at the sex differences in the daily changes underpinning social jet lag using an unobtrusive sleep monitor, which we can see here on the picture. This device has been validated against polysomnography for the assessment of sleep and wake and also of sleep stages. So this gave us the opportunity to also look into sleep stage distributions. And the study was carried out as an observational study of 156 Norwegian teenagers. And we monitored their sleep across up to 10 consecutive days. So to give some context to the findings on sex differences in social jet lag, I will first show the main effects of the day of the week, which we have adjusted already here for the effects of sex. In terms of timing of sleep, the teenagers went to bed later and woke up later on Friday, Saturday, and Sunday nights. In terms of the sleep and wake pattern, the teenagers had longer time in bed and longer time asleep, which is good, but we also found longer latencies to sleep and worse sleep efficiency on Friday and Saturday nights simultaneously, which kind of points to longer sleep, but somehow of worse quality because they take longer to fall asleep and sleep efficiency is lower. And in terms of sleep stage distribution, we observed that light sleep on Friday and Saturday nights increased at the expense of deep sleep, which decreased on Friday and Saturday nights. So then we looked into these interactions between day of the week and sex. And we found that girls, they fell asleep earlier on Friday and Saturday nights than boys. We also found that girls had shorter latency to sleep on Saturday nights than boys. And they also had, girls had higher sleep efficiency on Saturday nights than boys. And all of these results had large Cohen F-squared effect sizes. So these results led us to conclude that girls exhibit less severe social jet lag, especially in terms of overall sleep on Saturday night and the timing of falling asleep on Fridays and Saturdays. And this is in line with the studies based on actigraphic measures, but contrary to the studies based on subjective measures of sleep. We found no sex differences in sleep stage distributions across the week. And the major limitation in the study here is that we didn't have any other predictors of this association. So future research should explore kind of these mechanisms behind these associations. Relevant factors could be teenagers' mental health, bedtime procrastination, girls' menstrual cycles, stress levels, sleep hygiene, physical activity, chronotype, gaming, all of that. So that was a little bit about my research, and now I would like to take the last couple of minutes of my presentation to say a few words about being a woman in academia. first, a little disclaimer that I will speak from my own experiences that are based in a unique context. And to start with, I am living and working in Norway, which is considered as one of the most gender equal countries in the world. But despite that, there are still gender specific issues in Norway too, which include pay and pension gaps. There's fewer women in leadership positions and also a very gendered workforce in some occupations. And reports show that at Norwegian universities, about half of all the postdocs are women, while only 33% make it to professor level. And I thought, preparing for this presentation, that it was interesting to look into a little bit what could be the causes of this decline in career progression for women. And I didn't find much recent research done on this in Norway, but in Sweden, which is culturally very similar, they found that there are small but cumulative differences in the experiences of women and men in academia. Specifically, the report found that women experienced more unfairness and injustices in the system. Women had fewer opportunities for tenure, less access to mentors, and it was also more difficult for women to combine academia and family life. And so it is no secret that kind of making it in academia can be difficult for anybody with high workloads and job insecurity. But from my perspective and with the resources that I have been able to access so far in my academic career, I think it would have been helpful for women if in academia we had greater access to mentorship programs for women, for instance, which are country and field specific, because again, context matters. Then if the academic system encouraged representations of diversity to a greater extent, and also empowering women to take a proactive role in advancing equal rights and making the academic system more suitable for female leaders. So that was my 2 cents on that. Thank you for your attention, and please do not hesitate to get in touch on these platforms in case of any questions or comments.
All right, thank you very much. Thank you to all of our panelists. I really enjoyed your talks. I have some questions. I think I will work backwards, starting with Maria, moving backward to the previous panelists. Right, so Dr. Hrozanova, you did say in your next steps that you want to look at gaming. Do you think gaming and gender expectations about who a gamer is influencing this difference between weekend social jet lag in boys versus girls and screen time?
Yeah, I think, to be honest with you, I'm not, with maybe gaming specifically, there would be a gendered kind of expectations that maybe boys would be spending more time on gaming. But at the same time, I know that also among girls, it's these days it's a very common thing. And also maybe looking at gaming in isolation wouldn't make so much sense, because it's more screen time, I suppose, that also should be considered. And we know that teenagers these days are glued to their screens. So I think, yeah, this would be something certainly to look into the future. And I think there could be some pretty sound hypotheses expecting for there to be an effect. Thank you.
Okay, and the second question was, do you have any thoughts about school start times in the United States? And I know some places in Europe, we've begun to discuss limiting how early school can start for teenagers who tend to have a later chronotype, like to sleep in. Do you have any thoughts about that?
I think certainly later school start times would be helpful. In Norway, teenagers start at around 8 o'clock at school and have an 8-hour kind of school day, which means that they actually, if it was shifted a little bit, that would mean that they would get off school quite late. So I think this is the major factor that has been so far prohibiting the implementation of these later school start times. but it's absolutely something that would make a huge difference, I think, because in Norway specifically, in smaller places that are not in cities, kids often take an hour to commute just to get to school each way. So if they start at 8 o'clock, they need to be on the bus at 6:45, and then they gotta get up at 6 o'clock in the morning, and that's really yeah. I mean, it makes it basically impossible for them to sleep enough. So I think this is definitely something that would help.
All right, thank you.
Thanks.
My next questions are for Dr. Mong. So in your talk, you described giving gonad castrated males, ovecticized females, giving them the same hormones back, and then giving the same amount of estradiol to males. Did you try giving male levels of testosterone to the females?
Yes, actually, that we did do that. And in rodent brains, there's a very interesting enzyme. It's called aromatase. It's in our brains too, but more so in the rodent brains. And so what aromatase does is it'll take testosterone in a certain form, and it will convert it into estrogen. And so what we know is that when we gave testosterone to the females, it actually converted into estrogen, and we saw the same effects. Not as high, because partially that testosterone also went into something called a non-aromatizable androgen. So the second follow-up experiment that we did was we took the non-aromatizable androgen, dihydrotestosterone, that cannot be converted into estrogen. And when we gave that to females, we saw no effect. So androgens, pure androgens, do not change the sleep wake in the female brain. And I should also mention, we also did the study where we looked at the effects of progesterone. Progesterone is another ovarian hormone that sort of goes right alongside with estradiol in terms of the menstrual cycle, and then for rats, the estrus cycle. And progesterone had no significant effect on its own under physiological levels. Higher doses actually did increase sleep, but under physiological circulating levels, no change. And in combination with estradiol, there was no more significant change in the sleep pattern when both estrogen and progesterone were used. So at least in the rodent brain, we feel our evidence suggests that estradiol is the major gonadal hormone for female rats that's driving these behavioral shifts in sleep wake.
That's very interesting. Do you have any plans to look at other steroids or hormones that are known to be released differentially between males and females, for example, oxytocin or anything like that?
So right now we're not. I know that there's a little bit of work out there looking at other peptide hormones and the such. Where our work is focused now is trying to understand how estradiol in very specific sleep regulating areas of the brain is changing the neurotransmitters and neuromodulators. And so the one that we're really focused on right now is adenosine. So adenosine is that homeostatic barometer that tells us how sleepy we really are. And what we're finding is that, again, there are sex differences between the way that estrogen will modulate adenosine's effects. And essentially what estrogen is doing in the rodent brain is short-circuiting adenosine's actions by shutting down the receptors. The receptors will not signal properly, and in the male brain they do. They continue to work. So we're really excited about this new line of work that is looking specifically at the neuromodulators for sleep.
That's amazing. I had no idea that adenosine had any sort of sex differences.
Yeah,
We
we
have
were surprised too, and yes, so and we're not the only ones to see that. There's some other work showing that adenosine is also sexually sex differences in adenosine levels in the hippocampus and cortex as well.
wow.
So,
We have a
driven
question
by
from,
estradiol.
we have a question from one of our attendees. Would a feasible hormone therapy avoid the increase in insomnia and make a better program for women for sleep?
Yeah, so I have to be very careful how I approach this because I am a PhD, I'm not an MD, right? So, and there was a wonderful New York Times article about menopause last week discussing hormone therapy, so hormone replacement therapy. It is one of the most effective ways of treating menopausal symptoms that include hot flashes, insomnia, and cognitive decline. However, not all women can be prescribed hormone replacement therapy, but it is sort of one of those given in the right dose and at the right time, it is highly effective at treating insomnia and vasomotor flushes.
Thank you very much. Next, we have questions for Dr. Aggarwal. So in your talk, you were discussing cardiometabolic differences between men and women. Were there any differences in blood pressure dipping or how the blood pressure drops upon entry into sleep?
Yeah, so in the study that I led, it was actually a cohort study. So it was a prospective observational study among 506 women. So we measured blood pressure at several different time points. We didn't measure blood pressure dipping in the larger cohort. However, my colleague, Dr. St. Ange, who I work with at Columbia, she actually enrolled a subset of the larger cohort into a clinical trial to look at the effects of sleep restriction of an hour and a half per night for 6 weeks compared to healthy, stable sleep in the same participants. So it was a randomized crossover study. And she did look at 24-hour blood pressure. And so those results do show effects of sleep restriction, negative effects on blood pressure, dipping specifically, but I don't believe those have been published yet. So that is forthcoming.
And were there any differences between men and women?
So the study that we did was actually just among women, but we are planning to do this with both men and women so that we can look specifically at sex differences in that outcome.
Thank you. And the second question we have is, you reported that women were subjectively reporting poorer sleep and had increased inflammatory biomarkers. Do you think it's possible that the increased biomarkers were creating these feelings of sleepiness that led to them reporting worse sleep?
That's a really interesting question. And, it's something that we had considered, the bidirectionality of it. We're not really sure, to be honest, what was behind that. But I think that the key message for us as investigators was that, we have these objective measures of sleep and we were objectively measuring sleep duration. It's more difficult to measure sleep quality objectively, but there was something about their perception of their sleep quality that was linked to increased endothelial inflammation after adjusting for confounders. So there was something there going on, and we'd like to get to the heart of it, but we really feel that sleep quality has not been studied as much as sleep duration, and it's something that we'd like to continue studying, especially among women, to figure out what might be behind that association.
Okay, thank you. Our last set of questions are for Dr. Michalak. So did you find any differences in the benefits that men versus women received from napping?
So in our study, the biggest difference was in vigilance, and basically only in vigilance, because we also investigated episodic memory and spatial navigation and working memory. But basically vigilance was the one that showed difference, that women responded much worse to higher sleep pressure, but they really benefit out of the naps. So it was very interesting.
And then the second question is, you displayed some differences between the sleep architecture between men and women, where the men had more N2, women had more N3. Did you look at all to see if the amount of N2 was preceding N3 sleep or preceding REM sleep?
No, but also I look at the naps and we haven't had that much REM sleep because we had only 80 minutes. So we mostly had N1, N2, N3, depending on day or night. But REM sleep, we haven't had many episodes of REM in our study because of 80 minutes.
Right, yeah.
Long naps, yeah.
All right. Well, thank you all. See, I'm going to check, see if there's any. I think we've answered our questions. Thank you to all of our panelists for agreeing to speak today, and for speaking not only about your important research, but also your personal experiences. I think this is something that's very important for this society, because not only do we want to increase gender equality within our own society, we want to do good science, and accounting for sex differences and gender differences is something that leads to higher quality science. So thank you all for participating, and thank the attendees for coming today.